So I'm going to just give you a little overview right now of where our class sort of begins with studying bacteria. So the course is, again, microbiology. Microbiology is a study of really all like microscopic organisms, but really in this particular course we're focusing on bacteria. Although we will cover archaea, which is another domain. We will cover viruses to a certain extent. We're not really going into fungi very much. We'll talk maybe a little bit about that later on in the course. And there's all kind of protistan organisms. They're single-celled organisms, protozoa, that we're just not getting into, not in this particular course. It's focusing more on really prokaryotic cell biology, and that's kind of what we're going to start off with. What is a bacteria? So which is if our course is focusing on bacteria, we want to talk about bacteria themselves. What are they? Sorry, pen stopped working. Okay, so I'm going to move on to the next one here. What are bacteria? So a bacteria, the prokaryotic cell. So we're really focusing on prokaryotic cells. I have a lecture on prokaryotic versus eukaryotic cells, which you're going to follow. This one, which is going to go into more detail. This is going to be more of a broad introduction. In addition, all kinds of structures of bacteria I'm going to talk about now to a certain extent, but there are multiple other lectures broken down onto specific aspects of membranes and cell wall, different types of cell wall, extracellular structures like a calyx, the internal structures, inclusions and granules, and all sorts of other sorts of things that we find. So a lot of other lectures on the very specific details of this. This is kind of the overview to see before kind of getting into all that. So bacteria are prokaryotic cells, and hopefully we know what that means, right? So we know that karyo, right, means nucleus. Pro means kind of before, all right? So or no, so this is before the nucleus, and so these are cells with no nucleus, and that's no nuclear envelope, which is kind of a roundabout way. It's an internal membrane. And really, so prokaryotic cells, they're not just cells with no nucleus, they're cells with no internal membranes. No internal membranes. And what that's going to do, and I have another lecture on cell size a little bit and surface to volume ratio, is that having no internal membranes is going to affect the size limits of the cell. And again, other lectures that talk about the reasons, why in detail. What are the size limits of a cell? So for bacterial cells, there isn't just a size. There's not just one size, for a cell. In general, most of the bacterial cells are about 0.5 to about 5 micrometers in diameter. That's kind of an average. There are cells that are smaller. There are cells that are larger. The volume of bacterial cells is, in general, about 0.4 to 3, and this would be micrometers cubed, which is also femtoliters. So that's a volume. So then you might ask, what is a femtoliter? I've never heard of this before. Femtoliter is 1 × 10 to the negative 15th liters. So if you kind of picture so really the size of a bacterial cell, what is their size? How big is an actual bacterial cell? So think about it now in terms of volume. A liter, how much is that? So you think of a liter container of soda, or something like that. That's how much liquid there is. That's how much liquid there is. The volume of a bacterial cell is 0.0 10 to the negative 14 liters. That's how many zeros, 0.000000000, 14 zeros, and then the 1 there. So small volume. The other thing you're going to see is not much of a range in size. Maybe up to a 10 times range in size, roughly from smallest to largest. As opposed to, this isn't the lecture on eukaryotic cells, but as opposed to eukaryotic cells, again, eukaryotic cells can range wildly in size, usually going like 10 up to 100 millimeters, so 10 micrometers, to 100 millimeters, which is over 1,000 times. And that's just kind of average cells. There are extremes on both ends of those as well. So the bigger difference here to really take away, is that eukaryotic cells we tend to think of as being bigger. The bacterial cells that we study in this class are smaller. The smaller size we could keep almost kind of come with the largest bacterial cells and the smallest eukaryotic cells overlapping a little bit. But the eukaryotes get much bigger. It's not only that they're bigger, but they have a much greater range of size. And that, again, has to do with what limits them. What's limiting them is the membranes. Bacterial cells do not have the internal membranes, which means they can't create compartments with ions or certain chemicals. And so because of that, there is great osmotic pressure, and thus comes in the role of the cell wall. So bacteria are a type of prokaryotic cell with no internal membranes, and they're going to have a unique cell wall of peptidoglycan. And so you're going to need to know a lot about peptidoglycan structure, individual units that make up peptidoglycans, how they're arranged, and what are the differences in the distribution of them between what we call gram-positive and gram-negative cell walls. There's more to it than just peptidoglycan, but that's one of the big parts of it. So this is something unique. Plant cells, for example, have a cell wall, but it's not made of this material. It's made of other types of material. So this is something that's for the bacteria. Archaea have a cell wall, and people will say it does not have peptidoglycan, and technically it doesn't, because biochemically the molecules that make it up are different, but if you really study the chemistry of it, you'll find they're really very similar. The chemical differences are, in fact, in many ways very minor by comparison. So Archaea will have a cell wall. I have a lecture on Archaea cell walls and variation. There's actually all kinds of variations that go on there structurally and chemically. So the Archaea, another group of prokaryotes, has something similar to this type of cell wall, but the peptidoglycan cell wall is then unique for the group of bacteria, very specifically. So what do all bacteria have in common? Some of these things they tend to be small, roughly in this size range. They don't have the membranes. We will go into have a lecture on other structures, though. They have some kinds of internal structures. They're not just a sac with just liquid inside and stuff floating around. They have some organizational structure to that, and there are internal structures within the prokaryotic bacterial cells, but that's something else. So they can have that in common, but they can also be very different, in that particular way. In general, but the cell wall itself is going to be something common that they're all going to share. Other things that many of them have are some sort of extracellular structures. And those extracellular structures range from coatings, which largely are sugar coatings, different glycocalyx coating, capsule, but there can also be protein coatings as well, S layers, and a variety of other things that cover the outside of the bacteria. And then there's other sorts of extracellular structures that are extensions of the cell, such as flagella, fimbriae, and pili. And again, I have a lecture on those. What's the difference, similarities between them? Femtoleader and Pili, very similar to each other in terms of structure. Flagella are very different. These flagella are very different than they're the eukaryotic flagella that eukaryotic cells have, and how they're structured and how they actually work. So you'll need to go and learn the details of those later on. Not all of them have flagella. Some of them do not. We have motile and non-motile cells, so we kind of keep that in mind. Bacterial cells, in terms of cell structure, also have a certain cell shape, and the cell shapes are fairly limited, somewhat also by the structure of the cell wall. So we have cocci and bacilli, two very common ones, the most common ones that we're usually going to be discussing in the class. Those are the two main groups. We also have Spiruli, which kind of have this sort of shape. And then we have Vibrio, which tend to be shaped sort of like this. So the shape of the cell, is often unique, and it's sometimes a defining characteristic. One very early on method of categorizing and classifying bacteria would be first, cell wall, gram-positive, gram-negative, and then cell shape. So gram-positive bacilli, gram-positive cocci, gram-negative bacilli, gram-negative cocci, and so on. We're going to find that doesn't always hold true. There's a lot of variation. So a particular group that's related to each other could have cocci and bacilli in the same group and be really closely related to each other. But these are things that we use to help us describe and categorize them. So, and then these structures, these extensions, they would be, potentially they could be at one end of them like this. They could be surrounding them. It all depends on the bacteria and how those things are found. So those would be unique characteristics that we end up finding out for them. Another way in which they differ, from one another in many ways is going to be, their metabolism. So I wonder to use this guy here. So, as far as metabolism goes, the thing is here, we have, I talk about unique metabolisms, but in general, the metabolism of a bacteria is going to be, in a lot of ways, similar to our metabolism. So, they run glycolysis, pyruvate oxidation, citric acid cycle, have electron transport chains, same as our cells, which are really a lot of things that happen in our mitochondria, which we really go back to the idea of mitochondria coming from bacteria. So bacteria is a symbiote inside another cell, gave rise to the mitochondria over time. But that many bacteria have their own type of additional metabolic processes to produce other products through processes like fermentation. So they can produce alcohol and carbon dioxide and a variety of different types of acids and a whole number of chemicals that we know can be produced by bacteria. Bacteria can also get genes inserted in them, and then they can be used as almost a manufacturing plant to produce all sorts of things for humans. Even antibiotics can be produced from using bacteria. Bacteria can be used to produce them. We talked about the nucleus back here, and I guess I should kind of add to this. No nucleus, no nuclear envelope. The DNA, though, is circular. So, usually one chromosome. Typical. Not always the case. They can have more, but usually not. So most bacteria typically have a single chromosome. It is circular, so it's a piece of DNA, double-stranded DNA, and the two ends of the DNA are then joined together in a loop like this. And so that's a very common thing. That also helps protect it without a nucleus, since the DNA is in the cytoplasm, where other enzymes could potentially break it down if they could grab onto the ends, but now they can't because it is a loop, so they need specific binding sites, which might be hidden or protected based on other proteins associated with the DNA. these are some unique things about bacteria that we're going to be studying throughout the course. And the way the course is going to be broken down for us is going to be, we're going to start off really talking about structures, really to go through all the details of this part. We are going to then get into the metabolism leading to cell division in bacteria, and their reactions to different types of antibiotics and how the antibiotics work on them, essentially affecting aspects of their metabolism. Some of those things attack the cell wall, so that stuff then comes back and is related. We'll talk about the gene expression in bacteria, so the circular DNA, how it's replicated before the cells can divide for cell division. We'll also get into how genes are expressed. Bacteria do it pretty much how we do. DNA into RNA, RNA types, messenger ribosomal transfer RNA, come together to build polypeptides, which then get processed to become proteins. Same thing that happens in our cells as well that you would study in a just regular eukaryotic cell biology class. But they have certain variations. There's unique things that they're going to do because it's circular that doesn't happen the way that it does in our cells with linear DNA. So we can have some additional things, new strategies they have. They don't have meiosis for sexual reproduction to produce genetic diversity. So we'll talk about how genes can then be moved around so that we can add to diversity within bacteria. Types of gene transfer that they have, which is not like sexual reproduction, but it is a way of expanding and making their genome a little more complex, right? And so they can do things like pick up genes, from other organisms. Why do we study, bacteria? Well, there's originally disease. That was always, from the beginning, why anyone was ever interested in studying bacteria, was because of human disease caused by bacteria. Eventually, when germ theory was really realized and people had the tools to start to study these organisms, and Robert Koch was starting to isolate and identify organisms that cause disease, and then other scientists are able to study them, that's really the whole purpose of looking at them. But in addition to that, we now know that they have other importance. One is the microbiome. For us, the bacteria that live in us are very important to our physiology and our biology. So studying bacteria from that perspective, the beneficial perspective, becomes very important. The environment. For example, soil bacteria. Soil bacteria are extremely important for cycling nutrients, and their metabolism, some unique types of metabolism that they have, using nitrogen and a variety of different ways, producing certain products that make it available to plants as a fertilizer or food for the plants, that would only happen through processing with certain species of soil bacteria and certain types of fungi that live in soil. Those sorts of things become very important to all nutrient cycling in the environment. And things even like photosynthesis from cyanobacteria. So we don't realize that even the photosynthesis that occurs from these small organisms is significant, as well. And then there's commercial, uses. So bacteria can be used I mentioned they can be used to culture certain things. So bioreactors, these massive containers of bacterial cultures where the bacteria are essentially producing some kind of chemical that people are harvesting, and they drain liquid out with the bacteria and these chemicals, put more nutrients in so the bacteria keep growing at an exponential growth rate, but then they can extract out those chemicals from the bacteria that was producing them in that reactor, and those are the types of things, like I said, can be used in different types of nutrients and fertilizers, medications, antibiotics, all sorts of different products that can be produced by bacteria, and they're used in baking, in cooking and food preparation and developing cheeses, and a whole variety of other sorts of uses for bacteria in fermentation processes. So why study bacteria? Well, they really reach and cover all sorts of things, from basic human biology, human health. Some things we'll learn a little bit about as we talk about viruses, which is separate from the bacteria, but how much of our own DNA is actually viral DNA that has actually become part of us over time, and how it may have even affected human evolution is our association with different types of virus over time, and having their DNA inserted into ours has had an impact on our development as a species as well. So becoming very essential organisms in just the study of biology, in general, of human biology in particular, and then they have a whole bunch of links, to health and the environment. There are courses on environmental microbiology that really just focus on that particular aspect. There are medical microbiology courses that just focus really on the disease, aspect of it. Our course is a general microbiology course, so we're really covering a variety of these different aspects of it, but we're focusing on the organisms in general. So we're going to be breaking down the organism, looking at the structures, and then going through and looking at some of the processes, and then seeing the links to some of these different areas of biology that they affect us. So all these topics are then get broken down into other individual lectures, which you'll see following this one.